Mochel. He's our speaker today. He got his veterinary medical degree from the National Veterinary School of Alfort, which is in Paris, France, and then went on to DVM, which was in collaboration with the Collège de France, then worked on a PhD in Leiden University, which is in the Netherlands. And then he jumped the pond to become a faculty member at Iowa State University, my old stomping grounds. And more recently, he has joined us here at UGA in the Department of Pathology in the College of Vet Medicine. And he's leading the program in One Health here at UGA. And so, Jon, welcome, and please let us know about your work.
Thank you so much, Dr. Glenn, for the kind introduction, and good morning, everyone. It's great to be not physically you guys, but virtually to present some of our research in the field of computational sciences and comparative medicine. And so my job today, is and pharmacology can work together to create a tool that can be used for PMED applications, not in human medicine, but in veterinary medicine. So it's kind of a new and explored field thus far. But first, just a bit about my research philosophy when it comes to mathematical modeling. I'm pretty much a fan of the KISS or keep it simple approach. And so this was the cover of my PhD work a long time ago, featuring the bull by Picasso, which illustrates the fact that complexity in life can be captured with just 6 lines, right? So I mean, I'm a big fan of the simplest way to basically explain a very complex phenomenon. Now about the science, when you think about drug research and development, the current paradigm, and I'm sure you know that already, is well suited for human clinical trials. When you have the ability to enroll, tens of thousands of patients and to evaluate the effectiveness and safety of new therapeutic drugs. And that is something that we cannot afford in vet med, right? So all trials include at most hundreds of subjects, typically less than that, maybe 50 to 90. And that's also more complicated by the fact that we have an extremely large variability in the background and to differences in breeds and so forth. So we need to come up with alternatives to screen for innovation before going into the clinic in veterinary medicine. And that's precisely one of the objectives of our lab and this new Center for Precision One Health at UGA. And I'm sure you already know about the concept of One Health in the context of infectious disease and how interactions between the environment, the animals, and humans can actually facilitate the dissemination of disease between species. And that's our foundation, obviously, but we want to also expand on that concept to also include any kind of disease that can be shared between humans and animals, such as cancer, kidney disease, congestive heart failure, and so forth. So we believe that the information we can collect from animal patients, right, not animal subjects as experimental test case, can further our understanding of disease pathogenesis and contribute to the development of new therapeutic drugs for humans and vice versa. And that's what I think I would like to contribute to your student body, providing an experience on how these mathematical methods can help optimize the use of therapeutic drugs in humans and animal patients, and so exposing them to a broad and very large variety of problems which are centered around clinical applications in the end. So moving on to now our case study for the day. And so what I'm going to try to do and to actually, capture in the next 30 minutes is attempt to open the black box that stands between the optimal dose of a cardiovascular drug called an ACE inhibitor which is used for the treatment of congestive heart failure and therapeutic response. And that's a matter of of heavy debate in the vet med community, because the current recommended dose of that drug is 0.25 milligram per kilogram once a day, but practitioners in practice use a much higher dose, which can be up to 1 milligram per kilograms in total per day. And so there is kind of a disconnect between what has been, labeled and used for registration 20 years ago, based on some endpoints and some preclinical studies, and the reality of what practitioners do, because they actually observe a different kind of response in their patient population. So this work is trying to reconcile clinical data with mathematical modeling approach to kind of optimize the use of these drugs and their doses in dogs with congestive heart failure, because similar to humans, dogs also suffer from cardiac disease, as you may already know. And so to address this knowledge gap, we need to revisit two paradigms. First, the way we are approaching the biology of what we call the RAS, or renin-angiotensin-aldosterone system, which is this background biological pathway, which is target of this drug called an ACE inhibitor. And second, the methods we use to quantify the effect of these drugs, or the ACE inhibitors, on that biological pathway, which is called the RAS, or renin-angiotensin system. So we're going to start quickly with the biology of this pathway. And I don't know if you're familiar with the renin-angiotensin system, but it's kind of one of the most important hormonal system to maintain life, and to maintain blood pressure. And it's actually a fairly complicated pathway, which used to be, known to be centered around primarily two biomarkers, which we call angiotensin 2 and aldosterone. And for years, people have simplified that pathway and that cascade with those two biomarkers. And the way we have been developing these drugs called ACE inhibitors is basically to target that enzyme over there and reduce the production of angiotensin 2, which we believe is related to fibrosis in the heart and in different organs, which leads to congestive heart failure after many years of being overactivated. Well, practically this simplistic approach is just the tip of the iceberg. So what we know now is that there are multiple pathways of the RAS, and we typically use that classification between the conventional axis of the RAS, which revolves around angiotensin 2 here, which we discussed earlier, and that is linked to a series of negative effects, such as vasoconstrictions, hypertrophy of the heart, fibrosis, and so forth. And then an alternative pathway this time, which is centered around angiotensin 1,7 and 1,9, which exert an opposite effect to angiotensin 2. And so they have actually beneficial effect overall, including anti-fibrosis effect, anti-hypertrophic effect, and antioxidant effect. And so when you try to capture the effect of any drugs interacting with that system, what you need to know and what you need to do is to get a comprehensive overview of how much is this negative arm of the RAS, activated, so the angiotensin 2 here, and how much is the positive arm of the RAS with angiotensin 1, 9, and 7 also stimulated or inhibited with your medication. So I'm trying to use that analogy of, basically we have the dark side versus the bright side of the RAS when I explain that concept to my students. So unfortunately, most of the studies that were used 20 years ago to actually determine the dose of these drugs in veterinary medicine have been using that biomarker called ACE activity as a proxy for clinical efficacy. And so ACE was just representing how much, enzymatic activity I had in my system. And the goal was to basically, shut it down to almost zero to get to a therapeutic clinical effect. And what those studies have shown is that, you don't need to go very high with the dose of these drugs. If you go to 0.25 milligram per kilogram once a day, you already kind of max out the effect on that biomarker. And that was kind of the rationale for, this 0.25 milligram per kilogram once a day that was labeled when registered in the EU and in the US as well, actually. Unfortunately, we do know now that there is not a strict correlation between the activity of the enzyme and its product, which is angiotensin 2. So if I go back to this slide over there, you can see that ACE is responsible for Ang2 production, but inhibiting that enzyme over here does not lead to a complete blockade and depletion of that biomarker. And so there is a disconnect clinically between the level of ACE inhibition you see in patients and the amount of angiotensin 2 that they actually produce in their system. And in fact, we know that up to half of patients with complete depletion of ACE activity still have pretty elevated angiotensin 2 levels, which we know are, prognostic for morbidity and mortality in patients with cardiac disease. And so all of this to say that we need to kind of shy away from, walk away from those old biomarkers, ACE inhibition, to really capture the actual therapeutic effect of these drugs. And what we know from human studies is that two biomarkers, so angiotensin 2 here and aldosterone particularly, have been heavily correlated with clinical outcomes in patients with congestive heart failure. So we need to basically, look at those biomarkers more so than just looking at ACE activity as a proxy for clinical activity. Now, what I found to be very interesting is that, when you look at the information we get from, I'm going to call it the real world evidence of these drugs, is that clinicians already use, these ACE inhibitors at a much larger dose than the one based on ACE activity alone as a proxy of clinical efficacy. So as I was telling you in my introduction, practically the current recommendations is to use that drug for 0.5 milligram per kilogram twice a day versus the 0.25 mg per kilogram once a day. There's a fourfold difference between what was actually used initially from labeling studies and what clinicians recommend based on their clinical expertise. And so there seems to be some kind of a consensus between clinical practice and what we know about the biology of these drugs to maybe work on an optimization of these drugs based on more scientific evidence, basically. So now that we actually know about the biology of the RAS, I'd like to discuss some limitations of the current method that we actually use to quantify the effect of RAS inhibitors, especially in veterinary medicine. So maybe you remember this slide from the beginning. So in practice, what we do in vet med is we have kind of small study populations. So we have clinical studies of maybe 50 to 200 animals that we actually use and to verify the efficacy and safety of drugs in the clinic. But we also have to face a lot of inherent background variability that's really, makes our life more difficult to tease out a signal to noise ratio. So really finding an answer in the clinic is often difficult, especially when you rely on, very conventional statistics, because we're always very limited by the sample size of our cohort. And so, trying to make some generalization from a cohort of 50 or 60 dogs of certain breed characteristics to the entire population of dogs is extremely hazardous. And that's why, we're trying to push, for the use of more heavily computational methods such as MLs and mathematical modeling and simulations to kind of make a better job in our inference to make better decisions when it comes to doses in those dogs with congestive heart failure. So that's probably one of the the easiest way to summarize this information. And I wanted now to really, bring it home to this case study about the optimization of ACE inhibitors in dogs with congestive heart failure, and then break, through that black box that stands between the dose of an ACE inhibitor and the actual effect of that drug, on the RAS system. And the only way to do this is to look at exposure, right? So basically what we wanted to do with that study is one, characterize the exposure response relationship of ACE inhibitors on biomarkers which we know are relevant to congestive heart failure in dogs, which are Ang2, Ang17, and Ang19 particularly, and then build a simulator that basically would help us to optimize the dose of these drugs using different what-if scenarios. So I won't spend too much time on this slide, which is pretty crowded already, but just to give you a bit of an overview. So what we did is we actually conducted a study in healthy dogs, and we actually had three groups receiving different doses of NACE inhibitors using a partial crossover design. And so we used doses from 0.1 to 5 mg per kg twice a day, 2.5 mg per kg twice a day, once a day, sorry, as well in those dogs. And then we sampled them over time for looking at those biomarkers and the concentration of the ACE inhibitor in the blood of these dogs. And the overall goal of this exercise was to correlate drug exposure over time with the pharmacodynamic response we observed on the different biomarkers of the RAS, and then build a mathematical model related basically the drug concentration to the drug efficacy on those biomarkers to build this online simulator. So on top of this biomarkers, we also collected, different measures such as blood pressure and with some echo of the heart to check for the, functional changes over time as well. But really the majority of the work was focusing on collecting blood for characterizing the PK, so the exposure of the drug, and the PD, so the effect of these drugs on, on the renin-angiotensin and aldosterone systems of those dogs. The diagram of the mathematical model, the mechanistic model we built to PK of benazepril, which is the name of this drug, where on the different biomarkers of the RAS. So very, very briefly again, and I don't want to go into too much detail here, but this portion here that I'm highlighting over here is the kinetic model that we have developed. It's basically based on the binding model of the enzyme to its drug, which is to basically, modify the mechanisms and kinetics of the virus, then angiotensin, which are related to the production of angiotensin 1 and 2, in the blood. And then we had a very similar model for the tissue component of that system, because the RAS is actually found both in the systemic circulation and in different tissue compartments. So we had one model for the blood and one model for the tissue in equilibrium. And then we use different ODE systems to basically model the PK and PD of benazepril on those different biomarkers. So this next set of slides present the goodness of fit of the model based on the observation versus prediction plot. So as you can see, we have here the goodness of fits for the PK of the drug, so the kinetic of the drug for benazepril, right, in the plasma of those dogs. And then we had the same things for the various biomarkers, so which basically, again, translate the effect of that drug for angiotensin 1, 2, 1, 7, and then other angiotensins. And as you can see from that graph, we had a very good agreement between the model-based predictions and the actual observations over that populations of dogs. So we get some, consistency across the different biomarkers that the model was able to capture most of the signal in these dogs. Now looking at some individual fit this time, again, what we can see across the various biomarkers that we have sampled is that we were able to overall nicely capture the individual dynamic of the PK and of the response for these different dogs as well. So again, we were checking that box, and we are pretty happy with the performance of the model. And then, once we had confidence about the ability of that model to really capture the PK and PD of these ACE inhibitors on the various angiotensins, we knew we were actually able to build a simulation tool to really look at the relationship between dose, exposure, and response, and then optimize the dose of an ACE inhibitor that we can actually use in a patient population. So that's the front part of the exercise, the simulation step. And so now if you actually take a print screen of that QR code here, it brings you to an online simulator, which I'm going to open in just a second. So then we have the exact same thing all together. So you go. Simulations. All right, I'm going to reshare my screen again. All right. So hopefully this will work because the network is not great, but you should be able to see what I'm doing. So let's see. Share from here. All right. All right. So hopefully you do see my brother. And so if you actually look at here, you have different tabs, and one of these tabs is the dosage comparison. And so if you now look at here, you can compare up to 4 different dosing schedule. I'm gonna select maybe just 2. So that's here. The first one would be the actual currently labeled dose of this drug, which is 0.25 mg per kg for just once a day. And I'm going to give to that virtual dog 7 doses. And then the second one is the most twice a day this time. And again, I'm going to give it this time for 7 days, so 14 doses in total, because we give the drug in that case twice a day. Then you just click on run comparison over here. It's going to run those simulations in the background. And if we're lucky and the internet allows us, we're going to see now the results of those simulations. So that's what you see here. So the yellow plots are for the most extreme dose, like the high dose of 0.5 mg per kg twice a day. And the blue is a result of those simulations for the lower dose of 0.25 mg per kg once a day. Here you actually have the PK of the drug, which is the concentration time course for the ACE inhibitors. And then for the different panels over there, you see the dynamics of the response for the various angiotensins that belong to the renin-angiotensin system. So arm and the alternative arm. Placebo response, because we also had a placebo model embedded into our simulator, right? So what you can actually see here is that now you have different tabulated numbers. So we can calculate the area under the curve at different times, and then ask the program to calculate for us for the various dose the area under the curve versus the placebo for the 2 doses we have been investigated, and together with the 90% prediction interval from those simulations. So, and you can do that for all of the biomarkers. And if you look, for instance, for angiotensin 2, which we know is, I'm going to call it the bad guy, which is related to the negative effect of the RAS, we had about a 30% difference between the low dose and the high dose of the ACE inhibitors. Now, if I look at angiotensin 1.7, which is the good arm of the RAS, we had about a 50 to 55% difference between the low dose and the high dose. So those simulations are overall pretty consistent across the different biomarkers. And it seems that, if you actually go from the low dose to the high dose, you do have a significant benefit in terms of, inhibiting the arm of the RAS, which is angiotensin 2, and stimulating this time the positive arm of the RAS, which is angiotensin 1.7. So that's exactly what you want to see. You want to reduce the negative arm and stimulate the positive arm. And overall, you actually manage to do this much more effectively by using the higher dose of 0.5 mg per kg twice a day in those dogs. So now I'm going to go back to my PowerPoint. So I'm going to stop share here, and then I'm going to go back over here. And I'm going to do a share again. All right. So hopefully you do see my screen now again. So as I was telling you, we have three different panels for the simulators. One specifies the drug dose schedule. You play with how many dogs you can be up to, or actually downloading the figures from those simulations if you want to keep those for your records. And as I was telling you, we calculate the area under the curve, and we compare those between doses and placebo. So just to give you a bit of a summary and next step from this work, so what we actually saw was that the most robust response on the angiotensin 2, which is again the negative arm of the RAS, was really seen with the 0.5 mg per kg twice a day doses. Um, and, I did not run this simulations here for the sake of time, but if you were doing the exact same thing for the 0.25 mg per kg, but twice a day this time, not once a day, you had actually I'm going to call the middle dose. So the 0.25 higher dose when it comes to reducing angiotensin 2 concentrations in those virtual patients. For Ang17, same thing. The most robust response was also seen with the higher dose of 0.5 mg per kg twice a day. And as we did see for angiotensin 2, if we had run the simulations for the 0.25 mg per kg twice a day, we also had about a 25 to 30% difference compared to the higher dose. Also, a fairly close runner-up versus the high dose. So basically, based on those simulations, the 0.25 mg per kg uh, produced some consistent and more results, and they were overall superior to the current label once a day. So these simulations would actually with a clinical practice that basically physicians recommend to use these drugs and patients as well. But these are based from in healthy dogs on one
All right, so John was joining us from Europe. He was kind enough to be joining us from a long way away. So we'll see if we can get him back.
Oh, I see. I be this. Hello?
Oh, I think you're there. Yep, you're back.
Do you hear me?
We can.
All right, super. I'm sorry about this. I don't know when it stopped. Was it at the conclusion level, or did you hear the conclusions, or should I repeat that?
If you could repeat the conclusions, that'd be great.
Yes. Okay, absolutely. I'm sorry about this. I knew that there was going to be a problem with using the conference center network, but
No,
let's
you
see.
did pretty good.
Here
There
we
were
go.
only very few
All
times
right,
when
share
it dropped
again.
out. But yep,
Okay. I guess I kind of I broke the system with the simulations. It was just too much. So no, I was actually saying in conclusion that those recommended practice for the canine patients, which is the at least in that one of the very first example that we can actually reconcile empirism with background simulations. But it's these are being based only on healthy animals. And so, it's actually a bit of a risk on data from healthy animals. So what we're seeing in healthy dogs is data coming from clinical patients. So measuring the RAS of those dogs, in the clinic, and then fitting the simulator with actual data from clinical patients with congestive heart failure is the next step to make that tool more predictive. And we can also maybe make it a bit more fine-tuned to include some covids such as breed or stage of disease to make that really more of a tool for personalized medicine applications and be more specific to individual patient this time. And so what we're trying to do right now is to speak with the company who actually sells that drug and convince them to use those data to conduct a prospective study with different doses of an ACE inhibitor, and then to get data from dogs with cardiac disease, take blood from them, and then feed them all and make it more predictive moving forward. So that's kind of what I'm working right now with the drug company that sells the drug in the first place. And last thing, obviously, this is not only my work. It's been basically the work of one of my previous postdoc, Ben, who since then has moved to industry. So you take me if you join uh, the dark side of the of the force. I don't know, but actually he did a fantastic job over 3 years to get that project to conclusion. So with that, I'd like to thank you again for this, opportunity to present this work on using mathematics for treating a problem in dogs with congestive heart failure. It's been a very fun journey, and so what I'm trying to do with my students in the lab is really to expose them to a lot of different cases, which are very, very practical and clinically oriented, and then basically intrigue them as to how mathematics can basically be the bridge between science and clinics. And we are lucky that, or I mean, if we're lucky, but we are fortunate that most of these disease also exist in humans. And so we not only want to use those data to support, science for dogs, but also to support the way we're using these drugs in human patients as well. So we want to make it a One Health exercise also ultimately. So with that, I'd be happy to take any question. I would like to thank you for your attention.
Awesome. Thank you very much. If people have questions, go ahead.
Hey John, you know me. I think for
Yes.
this audience, you touched on this a little bit briefly, um, but you talked a little bit about kind of machine learning and biomarkers, but also like deterministic modeling, like ODE type modeling. I think, especially for this audience, the kind of general connections between these two, especially for Precision One Health, might be helpful. Because I think a lot of people in the audience they kind of do they work with various omics type data,
Yep.
or they do some sort of first principles based modeling. But I feel like it's often not both, or at least in those types of projects, often don't overlap. So I was just wondering if you could like talk about that dichotomy and how your guys work kind of bridges that.
Yeah, absolutely. So just to give you a bit of a background, I mean, when you develop a drug for animals, there is not any requirement from the air that you collect any data for PK characterization. So people don't really look about the exposure response of drugs to get a drug approved on the market in the. So, we actually are, and the level of quantitative science we have on those studies is fairly small. And so, what we're trying to do, with our approach is to say, okay, we're gonna take what is the best of the approach on the human side, and try to flip a bit the paradigm. And so, focusing on common disease in dogs and humans, such as cancer, for instance, We collect a lot of biomarker informations, both from preclinical dogs, clinical dogs, and in vitro studies as well. And we use a variety of approach, as Eugene was alluded to, such as omics or also obviously biomarker information, and to try to tease out, what is the relationship between the drug concentration we actually use and the effect we see, with a big question which is about extrapolation and scaling. So what we actually see, in a dish or in a dog does not mean that this is what you expect to see in an actual human patient, for instance. So, when I see a nice killing of a tumor coming from a dog with maybe osteosarcoma, and I would like to extrapolate that finding from dogs to humans, then the questions arise as to how do you scale this up? And so we also do a fair amount of between species extrapolation modeling to come up with ways to make sense of this data in the context of treating human patients. And so our lab is kind of I'm going to call it a mixed bag that combines in vitro systems, in vivo studies in healthy dogs, and in also diseased dogs from the teaching hospital that we have here in VetMed. And then using mathematics to kind of extrapolate those findings from those different situations to what we do expect to see in a patient that has the same disease, like a human patient in that case. So it's kind of an exercise of jumping across cell systems, animal models, and then species ultimately.
Yeah, and I think just to editorialize a little bit, a lot of us in IOB, we cover different model systems, model organisms. I think that cross-organism stuff that you guys do in the vet school could be an asset for helping us all collaborate a little bit more. So anyway, thanks.
Absolutely, 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 big time.
Are you able to see the chat, John?
Yes. So
Got a
chat, here we
question
go.
there.
All right, maybe. Okay, can you write examples on uniform balance? Yes, absolutely. So I can give you a very concrete example. So right now we are working on stem cell derived organoids, which are like, mini organs in a dish. And so we collect basically tissue samples from dogs with different diseases. And then we grow them, we grow either the tumors or the organs in a dish. And we do a lot of things such as RNA sequencing to dissect, the pathophysiology of a disease, or to look at the effect of a drug using different omics approach. And right now, I work with a student which helps us with that pipeline to analyze all of the data coming from RNA sequencing, and then understand what is the pathways involved in disease development, and B, what is the effect and what is the efficacy of different drugs we're trying to use to modulate those diseases in the first place. And obviously, in that exercise, we're trying to also look at similar pathways between dogs and humans, and then hit on different targets that we can use for drug development on both ends. So that's one example. The second one I can give you is we're working also on a program which is about Cushing's disease.
So we caught all of the first one. We're missing the second one here. I'm not sure if John can hear me. Oh,
I think he's gone again.